brain is built to generate movement. Okay, that's what behavior is. We generate movement. And we're just interested in understanding the brain.
Understanding how the brain coordinates movement is really a fundamental goal of neuroscience. Brains, in some sense, evolved to control movement. And all animals that have nervous systems or brains have to coordinate their movements. It's a process that can go from very simple reflexes to complex coordination of multiple behaviors.
The brain essentially cares about movement. Like what the brain does is move us around the world to find food, to run away from predators, to find a mate. So everything that we do in the end is coordinating movement. So to understand how that works is kind of essential.
There are a number of reasons we focus on the zebrafish model. Zebrafish, and in particular zebrafish larvae, are very tiny creatures and also completely transparent. So it's possible to see all the way through the brain in the intact animal. But although they're very, very tiny, and their brains are just about 0.5 millimeter across, they have many features that are shared with our brains and with the brains of all vertebrates. So we can look at fundamental aspects of computation in the zebrafish brain that can help us to understand computations that are going on in much more complex brains.
So what we were interested in is basically how the zebrafish transformed visual information into motion of the eyes and its tail. And what we wanted to understand is how neural circuits in the hindbrain, so a specific region of the zebrafish, participate in this transformation. We did this by basically imaging a large population of neurons and then understand from the activity of those neurons how that transformation from the visual input to the motor output happens.
The way we did this experiment is we took some zebrafish larvae, which are really baby zebrafish that are only 5 or 6 days old, and we put them in a dish where we restrain them with a jelly-like substance that's called agarose so that they cannot swim away. And so we put them under the microscope, that's called a two-photon microscope, where by shining a very powerful laser, focus on specific neurons that will fluoresce when the brain is active and see what they are doing when the fish is behaving. And we show them on a screen a stimulus that's either gonna suggest that the world is rotating around them or that they're drifting in a current of water. And we have a tiny camera that can look at the behavior of the fish so that we know whether the fish is moving the eyes or it's moving the tail.
The fish's brain is a three-dimensional structure that consists of tens of thousands of neurons. So to look at activity in the brain, we use a special microscope that lets us take virtual optical slices through this brain. So non-invasively, we record through all the neurons in one slice of the brain.
What I think was interesting is that we recorded this very, very large population of neurons, like 10,000 neurons. It's really complex. And using these, modern statistical methods, what we could sort of see is that in the end, it's actually quite simple. There are really only 2 patterns that the population activity was engaging in. One of these patterns corresponded to either leftward or rightward motions of the eyes. And the other pattern was representing basically when the eyes were moving to the front or to the back, or when the fish was moving forward, for instance. Okay, so despite the complexity that we initially envisioned, it turned out to be quite simple.
And then having identified these categories, we could look across different fish and say, what's the spatial distribution of these neurons? And something we found was remarkable was that the distribution of these different groups of neurons was very much the same from one fish to another. So there was an extremely stereotyped functional organization down to the single cell level in the brains of these fish.
People were debating, is the brain organized in such a way that there are like dedicated circuits for the left eye, dedicated circuits for the right eye, for instance? Or is the brain organized in such a way that there are dedicated circuits for, leftward or rightward movements of the eye versus, forward and backward movements of the eye? And so what we figured, it is really the leftward, rightward versus forward, backward that is the way the brain is organized in terms of these eye movements.
Traditionally, the way people have looked at the relationship between neurons and behavior was generally taking one neuron at a time or 2 neurons at a time and then trying to relate the activity of those neurons to the behavior. And what we think we bring with this method that we implemented here is that we take populations of tens of thousands of neurons and we use an approach where we can simultaneously understand what's happening at the whole brain level instead of neuron by neuron. And we think this is going to be useful for other people doing this type of experiments.
There are several ways that we'll want to follow up on this. So One is to make perturbations of activity using methods such as optogenetics, so where you can control the patterns of activity in neurons with light and to see whether they affect the behavior.
And there's one question that I'm really fascinated by, and that is throughout the brain, in many, many different circuits, we have sort of a dichotomy of cells. There are cells that excite others, and there are cells that inhibit others, so they just suppress the activity of other cells. And we actually don't understand that dichotomy very well. I think what we would want to do is sort of dive down deeper and understand in more detail how each of these populations works, and may therefore be in a good position to try to understand, how neural circuits compute at a more microscopic level.